I'll go over some of my results now. So first we'll look at stand competition or stand density between coastal and montane sites. So you can see on this graphic that the gray bars represent trees per hectare, so trees for a fixed area, while the black line represents basal area, so it takes into account how big those trees are, diameter at breast height. So you can see while on average coastal sites had less trees per hectare, those trees were bigger, while the inverse was true for montane sites where there was more trees per hectare, but smaller trees, so a smaller basal area. And then this graphic shows proportion of dead trees on a plot level. So the big blue dots represent, the dark ones represent plots which had over 35% mortality at the plot level. And so there wasn't a significant difference between proportion of dead trees between coastal and montane sites, but the average for coastal sites was an 18% mortality rate versus a 15% mortality rate for montane sites. And you can see the coastal mortality is kind of more concentrated down there. And then looking at mortality rates by species, so far we've concluded measurements. Brewer spruce had the least mortality as a percentage, only had 4% mortality rate observed, which is cool because it's a species endemic to this region. Whereas the pines had more mortality. Sugar pine had a 33% mortality rate, while western white pine had a 40% mortality rate. And then looking at mortality by moisture availability. So how we created these plots is we divided moisture availability into 3 categories for different species range in Northern California. So the x-axis is the moisture availability and y-axis is a percent mortality per hectare. So for white fir and Shasta red fir, we saw less mortality on the drier parts of their sites versus wet. And for sugar pine and western white pine, we saw the same trend with increasing mortality in wetter sites. So this kind of goes back to that first hypothesis, which says that maybe more mortality will occur in wetter sites. But this wasn't true for all species, such as the Brewer spruce, which had the inverse trend. And then looking at regeneration quantities by species, we saw the most regeneration in the fir species, Shasta red fir and white fir, and these are shade tolerant species, which mean they regenerate competitively in a low light environment. And these stands all were, fairly dense stands. And there's a pretty significant drop off between the firs and other species observed, with western hemlock and sugar pine being the 2 least observed species. And if you recall, we saw the highest mortality in sugar pine and western white pine, and they're both sitting lower ends of the regeneration quantity observed. My second research question was, did rates of tree mortality in Northern California and regeneration differ before, during, and after the 2012 to 2015 drought? And to answer this question, I had the same 54 quarter hectare study sites. And at each site, I cored a subsample of dead trees, and I used the cores to date when those trees died. And I chose the sample based on species and a subset of decay classes. So we divided the dead trees into 5 decay classes, with 1 being the least decayed and 5 being the most decayed. And so I was able to date trees in decay class 1 through 3. And you can see on this graph that there was a strong association, a significant association between decay class 1 and death dates during and after the drought, with less of a distinction between decay class 2 and 3 species. And in terms of how many of these decay classes I saw in the landscape, I observed the most decay class 1 trees, followed by decay class 4. So this kind of represents that pulse of mortality during and following the drought up here. And then I also dated regeneration. So how I did this was I sampled basal discs, or sampled the regeneration in the field, and used that basal disc to sand down to the root shoot interface and counted rings. Now I just think this graphic is kind of cool because it shows just how many rings can be packed into a little basal disc, which might be dime sized or smaller, or maybe a little bigger. So this graph shows year regeneration established and quantity on the y-axis. So the mean sampled height was only 14 inches, but the mean sampled age of the regeneration, so the small trees in the understory, was 37 years. And I just think it's kind of interesting to think about how small these trees are and yet how old they are. And just to put it in perspective, very few of the trees we saw were established during or after the drought, with most of the trees having been established in the understory prior to 2011 and some over 100 years old in the understory. So we hope that these data can help lead to enhanced forest management through more targeted silvicultural prescriptions, knowing the drivers of tree mortality, which species are dying can lead to more targeted management. And through knowing how trees are regenerating or maybe not regenerating, better decisions can be made to conserve the diversity of species in this region. And additionally, through having this fine scale information, we hope improved carbon sequestration models and other fine scale models can be built. So that's the end. I want to thank my advisors, Dr. Lucy Krahulis and Dr. Rosemary Sheriff, and everyone else who made this project possible. Dr. Eric Jules is also a committee member. Thank you so much. And I'm open to questions.
Yeah, Carol, I assume we have time for questions. Thank you.
I'm going to stop sharing so I can see everyone's face, but I can share again if there's a question about it.
Yeah.
I have a question. So did you personally go to all 54 of those study sites?
Yeah, I did.
Wow.
Yeah.
Without a helicopter?
Without a helicopter, no. We tried to select them so they were all manageable hikes in. Yeah, it was a super neat experience to be able to visit such a diversity of sites across Northern California and saw some really cool spots.
I have a question, Sophia. The mortality you're talking about are the trees, but I'm wondering if you have any sense for all those small trees that are part of recruitment. Like, did you notice any mortality in the seedlings, saplings, and even if you didn't analyze it, did you notice any pattern?
Yeah, I definitely noticed some. You know, it tended to be if I saw it, like if the stand was pretty diseased, or I noticed something was already occurring in the stand, there was more mortality in the understory. It wasn't something I'm analyzing, but I did notice that. But I mean, it's kind of, I think that's really interesting, which I didn't expect. I knew that you can't assume a tree's age based on its size, but I think it's pretty crazy that these regeneration aren't, I don't even know if I can call them regeneration. They might be the same age as some of the overstory trees, but yeah, I can't really speak. Less mortality in the understory in general, but.
So those older trees that are super small, I mean, I'm assuming they're just suppressed because they're not near a light gap. There's no light, and it'd be interesting to know if they could even respond.
I know.
Yeah. I
think that would be a great follow-up research question. Like, can these trees release? Like, if given the opportunity, are they viable options for the overstory? My just gut instinct is like, that would be pretty incredible if they released and could, but.
Yeah. Yeah, it will really govern what those forests become as you see these mortality events. You know, if those understory seedlings and saplings can't respond and get to the overstory, then whatever's going to replace those dead canopy trees has got to come in via seed. So yeah. Other questions? Okay. Well, thank you, Sophia, so much. That was wonderful.
Thanks for having me, everyone.
Yeah, this is great. This is nice to get an update from folks. Okay, well, I think Megan, you're going to go next. Megan Tygen is a graduate student in the biology department at HSU. She is working on a microbial ecology project. Her advisor is Dr. Catalina Cuellar-Gempeller, and she is going to talk about her thesis work, which is about the microbial ecology inside Darlingtonia pitcher plant, pitchers. All right, Megan, it's all you.
Thank you. Let me figure out how to share my screen again. Okay, cool. So, like Eric said, I'm Megan. I'm in the lab of Dr. Catalina Cuellar-Gempeller at HSU, and in her lab, we study host microbial systems, so large organisms like Darlingtonia and the microbes that live in or on them. My research is about bacterial communities facilitating prey item breakdown in the carnivorous plant Darlingtonia californica. if I can switch my slide. So Darlingtonia is the only known species in its genus, which is why we can call it Darlingtonia and not confuse it with any close pitcher plants. It's also the only pitcher plant endemic to the entire Western United States. It grows here in Northern California and Southern Oregon, and its close family members are Heliumphora, which is another pitcher plant in South America, and that family or that genus has 18 species. And then also Sarracenia, which is pictured here as B, which grows on the east coast of North America all the way through central Canada. And that genus has 11 species. So very cool family overall. Darlingtonia specifically thrives in moist, nutrient-poor soils. I have some pictures here on the right that I took. Serpentine fens are forested wetlands characterized by high metal contents caused by waters that flow through the characteristic rocks in these areas, and then they carry those contents into the soil. In addition, the soils have low nutrients available, so these characteristics make these areas pretty uninhabitable by a lot of plants, but Darlingtonia is one of the plants that have adapted to these conditions. It acquires its nutrients from insects and digests them in its pitchers down here. This plant has some pretty striking characteristics that sets it apart from a lot of other pitcher plants and makes it incredibly unique. So first, it has these downward facing openings. A lot of pitcher plants are open from the top, so this is very interesting. It also has these transparent fenestrations on the top of its leaves. On the right picture, these are the leaves, and then on the left picture, it is a flowering plant, so it has flowers and it has seed pods as well. But these transparent fenestrations on the top of the leaf, they're like windows. So when the insect flies in through the opening on the bottom, it gets lost and confused trying to escape through these windows, and then it gets exhausted and falls to the bottom of the pitcher where it's digested. And then Darlingtonia can absorb those nutrients. They also have this striking fishtail appendage, this little leaf, this extra leaf that grows off of the opening. This is interesting, and it's not really known what this is for. Some research has shown that the removal of this doesn't hinder prey capture, but there's not much else that's known about this. So all of this together makes it a very fascinating pitcher. Most related to my project is Darlingtonia is not known to produce its own digestive enzymes. So most digesting organisms like you and me, for example, we eat our food and our body makes the enzymes that breaks down the food in our stomach. And we have our gut microbes, of course, but we put in a good amount of work too. Darlingtonia, however, likely heavily relies on the microorganisms that live inside of its pitcher, that call its pitcher home, to help digest these insects and make those nutrients available. So to understand how the microbes do this, we need to understand a little bit about microbial diversity. Oftentimes, greater diversity coincides with greater ecological function. And when I say ecological function, I mean the movement of nutrients and the production of biomass. So oftentimes, low biodiversity will mean low ecosystem function, low movement of those nutrients, and then higher biodiversity will often coincide with higher ecosystem function, the higher movement of nutrients and production of biomass. Microorganisms specifically perform key functions in an ecosystem. So they recycle nutrients via decomposition. They break down dead plant and animal matter, and then they convert those into a form of nutrients that other organisms like plants and other bacteria can actually consume and use for growth. But what we find, interestingly, is that microorganisms don't always follow this high biodiversity, high ecological function trend. This concept overall is pretty understudied, but what we're sometimes finding in these systems is that high biodiversity in bacteria does not always mean the highest ecosystem function. And that peak might lie somewhere in the middle, but we don't really know. So for my research, I'm focusing on the bacteria in this system and on Darlingtonia, and I want to find the connections between bacterial diversity and insect degradation and how it helps this plant thrive. So in order to find these connections in Darlingtonia californica, I plan on taking 3 main measurements. That is chitin and protein breakdown. These are the key components of the insects that Darlingtonia is eating. And then also I want to measure nitrate and ammonia production because these are the forms of the nutrient that Darlingtonia can actually use for growth. And then lastly, I want to measure bacterial diversity. I want to know who's present in Darlingtonia, housed in that picture, and is breaking down the chitin and the protein, or who's producing that nitrate and ammonia that Darlingtonia is using for growth. I have taken these measurements in 3 groups. So first, I went out and sampled in the field over the summer. And for this group, I'll just be measuring bacterial diversity since I don't know how long the insects were in those pictures. I don't know what insects were there. I don't know the composition of the food they were eating. So I'm just going to measure the bacterial diversity for this group. For the second group that I'm growing right now, I have collected some of the bacteria from Darlingtonia leaves and I've grown it in the lab. So I will be feeding this culture insects, and at the end, I will then measure those nutrients I mentioned previously. And then I will compare what bacteria were present at the beginning and what bacteria were present at the end once that food is degraded. And then lastly, we have 15 Darlingtonia bacteria that were previously isolated in the Dr. Cuellar-Gempler lab, and I'll be testing these known bacteria on their breakdown and production of those key nutrients mentioned before. So before I get into my field sampling that we did over the summer, I just want to mention that this is part of a bigger project in the Dr. Cuellar-Gempler lab, looking at bacterial diversity in Darlingtonia across several fens in California and Oregon. So I had a lot of help for this and I didn't have to do all of this alone. So to sample these leaves for my field sample and serial dilution groups, first we snip a leaf at the base and they grow in these little rosettes, if probably many of you have seen, but we don't hurt the whole plant. We just take a leaf which grow and die on like year or 2 year cycles. And then we collect the fluid from the bottom of the leaf in a tube. This helps reduce the possibility of outside bacteria getting into our sample. And then we filter out the insects and debris. And then what we're left with is just a fluid that has the bacteria from inside of the pitcher. We can then freeze that fluid, which I did for the field sample group, and then I will be sending those in for DNA identification. Or if we want to use the bacteria in the future, we can add a preservative and then freeze it, and that allows us to revive the bacteria later, which we did for the serially diluted and the cultured community. Once we take those samples back to the lab, we can then extract that bacterial DNA, which is kind of a process, but basically physically destroy and chemically destroy the cells and then get rid of all those unwanted tissues. And then after many hours of work, we're just left with the bacterial DNA, which is different for each species. So we can then make a bunch of copies of that DNA using enzymes, and then we can send these in to be identified by an outside lab. At that lab that we send it to, they then break the DNA into a bunch of individual pieces, and a machine basically reads each of those pieces one by one. Then they send us a big old file of all those pieces, and we can match it back to a database that tells us exactly what bacterial species we collected. So I'm going to talk about our sample sites a little bit. The first site we visited was in Del Norte County, up here by Gasky. It's pretty easy to access, and possibly quite a few of you have seen it, but this is the nature trail right off of Highway 199. It's a well-traveled trail. Here's a picture from that site. Here's some of the Darlingtonia we saw. Some notable features of this site was that there was a good distribution of young, intermediate, and old leaves, and then cool and shady muddy water. The leaves were very big compared to what I'd seen at the greenhouse, some of them up to 34 inches tall. So I like to hold my arm out and like just imagine up to my elbow, because that's how big some of them were, and it was really amazing to see. The second site we visited was Shasta Trinity County by Mount Shasta here. So there was a stark difference between this site and Gasky. It was a big open meadow in the hot sun, and there were these two creeks running through it. And the Darlingtonia, of course, grew very close to the creek, that cold flowing water. Here's another picture there. We took temperature in the soil, in the water, and in the leaves to figure out what sort of what conditions these plants are growing in and what conditions the bacteria are surviving in. So some notable features of this site again was that a lot of these leaves were sitting, even though they had these cold flowing creeks, a lot of these leaves were in the hot sun all day. And that was a great contrast to Gasky. We found a lot of small, young, and intermediate leaves. These were a lot smaller than the plants we saw at Gasky. Our third site was Plumas County, and that's this third dot here down by the little town of Quincy. It was a hot, rocky mountainside. There's Darlingtonia up here. There's a pretty big fen. Darlingtonia over here, and then also here. Here's a close-up of that. So again, a lot of these were sitting in the hot sun all day, which was another stark contrast to Gasky, where we were in the shade. So it was super fun to get to work in the hot, hot sun all afternoon, sampling these. And these leaves were also pretty big, almost as big as Gasky, but Gasky was definitely the biggest leaves out of the fens we sampled. I just wanted to add my honorable mention site, the HSU Dennis K. Walker Greenhouse, where I collected samples for my cereal dilution. So I collected those bacteria and I'm growing them in the lab. And I'm hoping to be able to plot this on that same graph I showed earlier in the presentation, that biodiversity ecological function graph, with these dirty brown looking fluids being the most diverse and these ones being the least diverse. But I won't really know for sure until I send those in for sampling. And then here are the cultured bacteria, the 15 that were previously cultured in Dr. Catalina's lab. These were all collected directly from Darlingtonia pitcher plants. So as I continue onward with this project, I just want to say that this is very much a work in progress, and I'm excited to see what we find about bacterial diversity that Darlingtonia houses. I hope that I'm able to connect the field sample community and the lab simulated communities. We can create very controlled conditions in the lab, and I hope to see how much we can compare that to the unpredictable conditions we find out in the real world in these Darlingtonia fens. I'm hoping that this project can help us map the biodiversity ecosystem function graph in Darlingtonia so that we can better understand this fascinating plant and how it survives. And then on a broader scale, I hope that what I find here will allow us to explore and understand microbial diversity in other important California native plants as well. I want to thank the CNPS North Coast chapter for awarding this grant funding. It's been used for the kits that I'm using to measure nutrient breakdown and production, and also to help pay for the identification of some of the Darlingtonia bacteria. I also want to thank my advisor, Dr. Catalina Cuellar-Gempler, for her constant advice and encouragement, as well as my lab mates, Parker, Jonas, Taylor, and Travis, who helped me collect Darlingtonia leaves and proofread my rating. And last, I also want to thank the thousands of fruit flies that have given their life to help us understand how bacteria aid Darlingtonia in survival. Thank you so much.
All right, thanks, Megan. Awesome. All right, we definitely have time for questions. I want to ask a question, actually 2 questions. You don't know how many bacteria species are in the pitchers yet, but what's your guess? How many species do you think is in an individual pitcher?
Previous work that Dr. Catalina has done, she's collected hundreds of species within one single pitchers. So
yeah,
it's pretty intense.
And are they species that are known already or match other, studies, or are they new?
There are a good portion of them are known, have been collected, have been identified elsewhere, but some of them aren't known. And the best that I can get from the database is some of them just don't match anything that have been found. And some of them have a close relative that can be closely matched. So some of them can be matched by genus, and then some of them just say unidentified.
Okay, I have one last question from me. What's your guess in terms of how much variation you're going to find between pictures that are in the same fen and then between fens? Like, are the pictures different from each other in terms of the microbes, and are the sites Quincy really different than the Smith River, or are they the same?
I definitely think Catalina will know more in a few years, because that's part of her bigger project on the Darlingtonia fens across California and Oregon. But my guess would be that there would be quite a lot of similarities between the different fens across large spatial separation, because these plants and their bacteria co-evolve together, and hosts sort of select the microbes that live on and in them as well, because there's some sort of optimal combination that helps the host and the bacteria survive.
Great, thank you.
Yeah, thank you.
Any other questions?
I have a quick question.
Yeah.
I was wondering, in your Shasta County site, was that Deadfall Meadows?
It might have been. I don't remember the name.
Okay, cool.
I just remember it by I don't even remember. There was like a road sign, but I don't remember what the road sign said. I have a picture somewhere in my phone.
I'm wondering if you're expecting to find different kinds of insects from the shady place to the sunny places.
Yeah, we actually collected the insects. So we separated the debris and the insects, but we collected those in separate tubes. So maybe, hopefully, if we can find someone who specializes in insects in our lab, they'll be able to do a little more analysis on that. But in the sunny sites, I definitely found a lot more like big bees, wasps in those pictures. Whereas in the shady site, Gasky, there was a lot of little mosquitoes, little flies, little beetles. So I don't have any exact numbers on that. That was just my personal observation.
I do have one more follow-up question that just reminded me of. So these fens rely on fire. So what happens if you don't have fires? They get encroached by woody plants, they get too shady, and then eventually the Darlingtonia can't really survive under the canopy, as well as a lot of the other fen plants. So fire is kind of a reset for a lot of these fens. And the Darlingtonia generally don't die in a fire. You just lose all the above ground vegetation, and then they put up new pitchers the next spring. And I was just thinking about that. Where do you think the bacteria come from when a fen burns and then you get these brand new pitchers and all of a sudden they have to start over again? I know you don't know the answer to that exactly, but just any guess how the microbes restart?
I think so. What we know about Darlingtonia, especially when the pitchers first open, is that we kind of hypothesize that's a pretty sterile environment. We don't know for sure. Maybe it's getting those bacteria from the water that it's drawing up in its roots, or maybe it's a pretty sterile environment in there. But I would guess that a lot of the microbes come from the insects that fall into the pitcher. And another interesting concept in that is that only around 90% of the insects that fly up into the opening, 90% find their way out. So less than 10% actually end up falling in the pitcher and getting eaten. So a lot of those insects are going in there, adding their bacteria, and then they leave.
Yeah, okay. Yeah, that's cool. I hadn't thought about that. That makes sense. Thanks.
Yeah, thank you.
Yeah. Any
Those are super fun
last-
questions.
Yeah, no kidding. I love Darlingtonia. Any last questions? There's other Darlingtonia lovers in this audience, but they're being quiet. Okay, well,
Thank you,
thanks,
everyone.
Megan. Yeah, thank you so much. All right, Sarah, you're going to be next.
Hey,
Uh,
Eric. I'm
Yeah.
a Darlingtonia lover. It's Lisa Hoover.
I know, I saw your name down there. I was expecting you to
I
say
know,
something.
I know. Sorry, folks. Megan, just make sure the Six Rivers National Forest, keep them in the loop on what you find out, so that the botanists there can take this forward and apply it to conservation and management. Thank you very much.
Okay, thank you.
All right, thanks, Lisa. All right, so our last speaker is Sarah Norvell, and she is also a graduate student in the biology department at HSU. Her advisor is Dr. Terry Henkel, and I see Terry is also here. And Sarah's project is just one of these wonderful projects that I wish I was doing. She's doing a lichen flora of Horse Mountain. So everybody loves and knows Horse Mountain. It's all yours, Sarah.
Thank you, Eric. Now I'm excited to share some lichen info with some plant people. Okay. Can everyone see my screen? Perfect. So like Eric said, my name is Sarah Norvell, and I'm a biology graduate student at HSU, and my graduate thesis is a macro-lichen inventory of the Horse Mountain Botanical Area in Six Rivers National Forest. And before I get started, I just want to thank the California Native Plant Society, especially the North Coast chapter, for having me here tonight, and mainly for providing me the opportunity to do all of my field work this season. And they're also providing funding for my DNA sequencing that will happen in the spring. I have also received funding from the California Lichen Society and the Oregon Mycological Society, all of which has been greatly appreciated. My graduate advisor, Dr. Terry Hinkle, along with my graduate committee, Marie Antoine, Dr. Eric Jules, Dr. Heidi Rutschau, and Dr. Bruce McCune have all provided invaluable information for me over the last year. I'd also like to thank Six Rivers National Forest for all of my collecting permits and for letting me tromp around up in the botanical area for the entire summer. And then I've received a lot of lichen help from Tom Carlberg, who is the president of the Lichen Society here in California. And I've received a lot of help from a couple of my colleagues, Adrian Kovasi and Caitlin Alchin, and as well as my trusty field assistant, Hugin, my dog, who typically it was just the two of us up there all summer in my plots, and he deserves some recognition for suffering through lots of chaparral climbing with me. As for my thesis objectives, I started out by intending to survey the Horse Mountain Botanical Area to collect all species of macro-lichens present in order to provide Six Rivers National Forest with the most accurate species checklist possible. And I have completed my fieldwork portion. And I'm currently working to identify all of the specimens that I collected, and I will be curating extended voucher specimens of all the species that I encountered. Extended voucher specimens just implies that I will be making these little lichen packets, just like we use in the plant herbaria. We have lichen area that contain lichen specimens. And by extended, I just mean that I will be entering all of these into a database, digitizing them, taking photos of them, adding all their metadata, so that they can be accessed by lichenologists all around the world. And then I will also be obtaining the ITS barcode DNA sequence for each of my voucher specimens. This is essentially just a short little DNA barcode that's used for fungal species identification, and it's also been proven to be successful in lichen species identification. I'm actually going up to Oregon State University next week to meet with Dr. Bruce McCune, who is going to show me all of his lichen PCR protocols so that I can do that on my own in the spring. And then finally, I want to compare my plot species richness and community composition with other lichen inventories around the globe, mainly as a bonus just for my own edification. But I'm curious to see how my inventory compares with those that are also conducted on serpentine. Serpentine was one of the main reasons that I chose the Horse Mountain Botanical Area for my inventory. You can see here in this little map that I stole from Backcountry Press and Michael Kaufman, serpentine outcrops in Northern California are in dark gray here. And just west of Willow Creek, you can see the Horse Mountain Botanical Area sits right on top of one. And as most of you know, serpentine outcrops tend to host pretty unusual plant communities due to the chemical makeup of their soils. So on Horse Mountain, I'm primarily interested in Pinus jeffreyi. They typically don't grow so close to the coast, but these tend to enjoy the harsh serpentine soils. And then Chamaecyparis lawsoniana, the Port Orford cedar, experiences the southern extent of its range within the Horse Mountain Botanical Area. So because a lot of lichens are epiphytes, it follows that an unusual plant community might host an unusual lichen community for the area. And because you're all plant people, I'll pander just a little bit. These are two of my favorite plant species that I encountered while I was up there this summer. This is Lilium pardalinum bulmeri, Bulmer's lily, and this is the Alaska piparia, which is Platanthera unilacensis. Additionally, because you're all plant people, I thought it would be most appropriate to give a very brief introduction to lichens, ever so brief. And I will also include some basic lichen terminology so that maybe you all can go forth and start identifying lichens on your own if you haven't started already. I will also show you what my plots look like and how I'm able to go about surveying for something so small in such a large area. And then I will finish up by sharing just a few of my favorite species that I've identified so far during the inventory. And hopefully we'll have a couple minutes left over for questions. So what exactly is a lichen? Lichen is short for lichenized fungus. So essentially a lichen is a fungus that has evolved a complex nutritional strategy. So instead of obtaining their carbon via decomposing decaying matter or by forming associations, say via mycorrhizae with plants, lichens capture free living green algae or cyanobacteria in order to form a lichen symbiosis. And the free living green algae and cyanobacteria are able to photosynthesize. So the lichen forms this nutrient exchange system around what we call the photobiont, the photosynthesizing partner, and is thereby able to uptake carbon from the photobiont. We call the fungal part of the symbiosis the mycobiont, and they form this new stratified structure where you get a layer of the photobiont surrounded by fungal hyphae in different forms, they end up being ecologically stable mutualisms. So the photobiont, like I said, they're free living. There are only about 200 species of photobiont in existence, whereas there are upwards of 20,000 species of mycobiont. So these photobionts get shared among the mycobiont species. So when we talk about a species of lichen, we're actually referring to the mycobiont because they are obligate, they cannot exist without capturing that photobiont. They have no other way to obtain their carbon. And the photobiont actually does get something in return. The mycobiont is providing first a structure, so a little microclimate for that photobiont to exist in. And the mycobiont also, just because of the nature of the fungus, it's able to uptake mineral nutrients and water just directly across its surface. So the mycobiont provides mineral nutrients and water to the photobiont in exchange for carbon. So you've heard me mention that I'm doing a macro-lichen inventory. What does macro-lichen mean? We'll take a look at this picture here. I actually took this through my hand lens. This is a very tiny rock-dwelling lichen. You can see that these lichen thallus, the lichen body lobes, are no greater than 2 millimeters wide, so it's very tiny, but it is still a macro-lichen. Macro-lichens, by definition, simply have both an upper and lower surface that is separate from its substratum. So this macro-lichen can actually just be scraped right off the rock and the bottom part of the thallus is still intact, as opposed to micro-lichens, which are fully embedded in their substrata. If we zoom out of this picture a little bit, we can see this gray stuff in the corners. To the untrained eye might just look like rock, but that's actually a species of micro-lichen that's fully embedded in the substratum. So in order to identify that micro-lichen, I would need to chip it out of the rock and then put it under a compound microscope to look at the spore structure. So micro-lichens take a lot more time to identify. I think if I were to include micro-lichens in my Horse Mountain inventory, it would probably take me like a decade or so. So needless to say, I will be focusing on the lovely macro-lichens instead. Again, those which I can just remove from their substrata without damaging the lichen thallus. So I want to just give you a brief overview of lichen body plans so that you can start to identify them on your own. If you are going to do this in Humboldt County, I highly recommend this book, Macro-Lichens of the Pacific Northwest by McCune and Geyser. It has great keys and photos. And I know we're not technically in the Pacific Northwest, but it works great for Humboldt as well. So starting on the left here, this is Ramalina menziesii. This is our California state lichen. And this is a fruticose lichen. So it doesn't have a clear upper or lower surface. It's kind of shrub-like, as opposed to this foliose lichen here. I like to remember it by thinking of like foliage, a leaf that has a clear upper and a lower surface. And then down here in the bottom right, this is what we would call a squamulos lichen, kind of a funky body plan, where they have these small scales, like little overlapping flaps called squamules that make up this squamulos lichen. Of course, this is Cladonia, which is pretty well known. They have these additional little cup-like structures. We won't get into that. But anytime you see these little flaps, that is a squamulos lichen. You will also see crustose thrown around in the keys. They are referring to those micro-lichens when they're talking about the crustose lichens embedded in their substratum. There are several morphological traits that we also use to identify lichens, primarily their reproductive structures. On the left are their sexual reproductive structures. These are called apothecia, singular is apothecium. They are giving off fungal spores. So the fungal spore will still need to recapture the photobiont when it starts to germinate, but they are able to disperse long distances with these little fungal spores that are microscopic. And these apothecia differentiate a lot across different species. You're able to identify them just based on characteristics of the apothecia. So you'll run into those a lot in the keys. And then you'll also look at Ceridia and Isidia. These are the asexual propagules. So these little Ceridia are essentially the symbiosis in a small package, just ready to be sent out into the world as a new lichen. They look really powdery on the surface of the lichen, just because they're little groups of fungal hyphae wrapped around photobiont units. And then the Isidia are just a slightly more advanced version of those Ceridia. They also contain both parts of the lichen symbiosis. They just need to be broken off of the lichen, and then they can start as a new individual. They already even have the tough outer cortex layer, the upper surface of the lichen. Marie Antoine describes these as shiny little sausages, which actually is very true if you look at them under a scope. So how do I go about identifying lichens when I'm in a place where they look like this? I took this picture up on Horse Mountain under a Doug fir, and it's really intimidating at first, but just like all of you have experienced your green blurs kind of coming into focus with plants, that can totally happen with lichens, and probably much quicker than you think. There's a lot of repetition of species in these huge clumps, and it becomes pretty easy after a while to just scan branches as you walk around and look for unusual species. So I followed the FIA protocol for all of my plots. This is the Forest Inventory and Analysis done by the US Forest Service. They use these FIA protocols for FIA plots for tons of different organisms. This gray circle here represents the lichen plot. And I don't worry about all these little subplots. They don't actually apply to lichens. I just use them to kind of help orient myself when I'm sinuating through the plot. So I use the FIA protocol so that I can compare my lichen inventory to a ton of other lichen inventories that also use this protocol, so that we have the same time frame. You're on a 2-hour limit, and you basically just sinuate through the plot, slowly collecting new species as you encounter them. I have a little bag of lichen packets I walk around, and I put a new species in the packet and label it as best I can based on my field ID. And then I will record the substratum and an abundance rating that's also provided by the FIA protocol. And then I take all of those specimens back to the lab for a look under the dissecting scope and for chemical spot testing, et cetera. It's important to note here that the FIA plots only look at epiphytic lichens. So I kind of split my plots into 2 different surveys where I did 2 hours for the epiphytes, and then I would come back and do 2 hours for the ground and rock lichens that I was also interested in for this survey. So you may be thinking that lichens seem a little bit complicated and maybe complicated to ID. Why should we even bother with them? We should bother with them because they play hugely important ecological roles. They dominate 7% of the Earth's surface and provide critical nutrient cycling in many of our ecosystems. And they also provide nesting materials and winter forage materials to a lot of our favorite organisms. A lot of birds use them as nesting materials because they're antimicrobial. And then lots of small rodents additionally use them for nesting. And then winter forage diets by elk and caribou and reindeer. Lichens are really important for those. The flying squirrel locally, 90% of its winter forage diet is made up by one species of lichen. So they are absolutely dependent on lichens to make it through the winter, as are these little tree voles. And guess who is dependent on the tree voles and the flying squirrels? Our beloved spotted owl. Countless arthropods also rely on lichens. Many have evolved to camouflage with lichens. You can see this little moth that I'm outlining here. And then there's a spider in this lower left-hand corner. Many spiders inhabit lichens, as do little mites and springtails. The springtails are actually pretty cool because they ingest some of those lichen propagules that I was talking about. And the lichens can survive their digestive tracts. So they actually end up dispersing the lichens a little bit as well. So understanding and appreciating lichens leads to the conservation of lichens, which leads to the preservation of a lot of biodiversity. So now I just wanted to share with you some of my favorite species that I've encountered so far up there. I had not met this one previous to this inventory. This is Atiana sphaerosphaerella, this kind of yellowy, creamy colored lichen. It actually looks a bit like it's crustose, but it's just really closely oppressed to this branch. And then it looks like there's a lot going on here, but really I only see 3 other species of lichen. This is Hypogemia imshaugii here. You can easily identify Hypogemia because it's the only lichen that has hollow lobes. And then this little scraggly fruticose thing in the background is Notobryoria oregonensis. You see a lot of that up there. And then this brown foliose lichen, it's really warty on the surface, is Cetraria Platyphyla. So we see a lot of that up there. So it's really easy to start breaking down these branches once you just can recognize a few species. This Atiana I also really like because we use UV light sometimes for lichen identification. And this one fluoresces this gorgeous like brick red color. Vulpisita Canadensis is one of my favorite little sunny species you can run into up there, bright yellow because of the volcanic acid that it contains. It is foliose, while you might confuse this Litharia columbiana with it if you're first starting out, just because of the colors of the apothecia. But you can see that the Litharia is much more fruticose in nature. Same volcanic acid causing the similar color. These are both really easy IDs if you're up on Horse Mountain. This is one of my favorites. I love this lichen. This is Seroma hypnorum. It's really teeny. I took this one also through my hand lens. You can see these little baby moss leaves. These are probably just 1 millimeter or 2 wide. And I just love the way, this is a squamulos lichen. You can see the little squamules. And I love the way that the squamules continue around the edges of the apothecia. It kind of looks like they're got little pearl necklaces on. And then Normandina pulchella, this actually always shows up as an incidental lichen, which is what happens when I bring a lichen under the dissecting scope and realize that there are actually lichens living on that lichen. So this brown background is a different lichen, and these teeny little things we call elf ears, the common name of this lichen. You can see the seridia, the powdery stuff here. Those are those little sexual propagule or asexual propagules. This is just a really fun one to find by accident, really. And then we have Umbilicaria Angulata. I had also not met this lichen previous to the inventory. They have these amazing apothecia that are in little angular kind of triangles. And they have concentric rings and folds to increase the surface area of their spore producing surface. Just like a mushroom has gills, same concept, the Umbilicaria, which is a rock lichen, has these amazing little apothecia. If you were just looking at it from far away, it would just like look like little black dots. These are also probably only about 1 millimeter wide. So always pays to look a little bit closer. And then finally, this I just think is really cool. You get some just basic lichen species, but after a while, you start to realize that you recognize when the lichens look odd. And if you see here, this really weird bulbous thing is actually a lichen gall that's been caused by what we call a lichenicolous fungus. So that is a lichenicolous, that's a fungus that has evolved to parasitize lichens. And so it's created this little gall structure to reproduce on its own. You can see all these tiny black dots are actually the reproductive structures of the lichenicolous fungus. Pretty crazy. And then I just added this Ochroletia here. Again, this is a micro-lichen, but this is a really easy one to ID. They all have these little, what Marie Antoine calls pumpkin pie apothecia. This is the pumpkin pie lichen. And so if you go up to just about any tree, you can look a little bit closer and try to find these little baby pumpkin pies, and you will know that you have found Ochroletia. So thank you so much for listening. Here are some of my sources and photo credits. And I appreciate you letting me go a couple minutes over.
Awesome. Thank you, Sarah, so much.
Thank you.
Yeah.
I'm happy to
So-
answer any questions if there is any time left.
I'm sure we have time for a couple questions. Can we, Carol? Yeah, okay. I was gonna ask you who took all the pictures, but you answered that right at the end. So you
I took
some
most of them. I didn't take all of the animal pictures, of course, but most of the lichen pictures were.
Oh, okay, okay. Yeah, well, they're really good.
Thanks.
Yeah, my one real question was, even though you might not know exactly yet, I'm just kind of curious if you have an estimate of the number of species that you think you're going to find and
Sure.
how many per plot on average.
Yes, so I collected roughly 40 specimens per plot. So I ended up with about 800 specimens. And I've so far discovered about 60 species, but I really haven't gotten into my jelly lichens. And I know we didn't talk about that. Those are extra difficult to identify, and I'm hoping that PCR will help me with that. So I'm hoping upwards of 60, probably 75 or so total.
Total. And is that a number that you're surprised by, or that's about right? What is Bruce?
It's about right. I think I was thinking about 50 when we first started out. And just from walking around up there, I was thinking 50, if not a little bit less. So it's been nice to discover some of those smaller moss-dwelling lichens that I wasn't expecting, and quite a variety, quite a lot of diversity in the jelly lichens that I'm hoping to get into soon.
Cool, cool. Yeah. Any other? Carol is raising her finger.
So now that you've been up there looking at lichens, are you seeing different lichens on the serpentine than when you're not on serpentine?
That's a great question. I think that it's not so much about the serpentine, but I definitely do see a difference when I'm, say, in the like more shady Doug fir areas on the western side of the botanical area, as well as that there's a bunch of Quercus vaccinifolia over there, and the lichen communities in that area are entirely different than the ones that are among the Chamaecyparis and the Jeffreyi, the Pinus Jeffreyi, and the sugar pines and all of that. I got to the point by the end of my plots that I could really expect certain species to show up depending on what plant community I was in. So I found that the plant community seems to be the driver more so than the actual serpentine itself. I haven't found any serpentine rock lichens that have really surprised me yet.
Interesting.
And that seems to be consistent with the literature so far.
Very cool. All right.
I don't want to hog the airwaves, but
Go ahead.
this is a lichen question that came to me, as the contact person for our chapter. This woman from Southern Humboldt phones and says, well, she called it moss growing all over her oak trees is killing her oak trees.
No, it's great for the oak trees. It's an interesting dilemma because typically when trees start to die, they end up, there's less shade in certain areas, so they have more sun exposure. So typically lichens will come in after the tree is already suffering from something and inhabit that area of the tree. And so then people think, oh my gosh, it's the lichen is taking over and killing the tree. But really, if anything, the lichen is highly benefiting the tree via nitrogen cycling. It kind of acts as a little sponge, just like bryophytes do, to hold water from rain over time and kind of slowly just distribute it to the tree as opposed to letting the rainwater just run off. So if you have lichens on your fruit trees, keep them. Do not buy any of the junk out there that tries to kill lichens on your fruit trees. They're always beneficial.
Great. Lisa, you have your hand up. Your turn.
Thank you, Eric. Hi, Sarah. Lisa Hoover.
Hey, Lisa.
Here. Hi. Yes. Thank you so much for pointing out the ecological biodiversity network of lichens and the role they play. And you mentioned early that you were looking at Jeffrey pine and Porterford cedar. In your collection, is there any substrate specificity? Because we have Western white pine up there and incense cedar and things like that. And so I was just wondering, when you provide us with the 6 rivers with that amazing list for the Horse Mountain, I'm curious about the substrate. And it was interesting also to learn, because I thought there might be some endemism associated with maybe lichens and also the chemistry of the ultramafics that might influence that flora, but it doesn't sound like that's the case. So thank you. And yeah, the substrate association is of interest to me. Thank you very much.
Absolutely. So for each specimen that I collect, I note its substratum. I note the species of tree if it is on a tree. I'll even note if it was on the tree bole versus the tree branch. And then yes, I certainly note the tree species. So hopefully that will come into play when I'm doing comparisons later on. I might be able to see some patterns there. But of course, the lichens don't necessarily need to be attached to a particular tree species because they're obtaining all of their nutrients just across their bodies. It just tends to work that way based on the pH of the bark. Some lichen species are better suited for certain pHs than others. And so you tend to see certain lichen species on certain trees like oaks versus conifers. But that's pretty much as far as it goes.
Thank you.
Thank you. And thank you for helping me get my permits.
Yeah. All right. Well, thank you so much. All 3 of those talks, they were fantastic. Sarah, Megan, Sophia, really, those were 3 great talks. Thank you so much. I'm going to hand things back to Carol to wrap things up.
Okay. Yes, thank you. They were really very professionally given and interesting stuff in all 3 of them. You guys can be proud, and we're proud of you, knowing that we had a little bit to do with it. So I guess we can call the program officially over, but if anybody wants to hang around and talk like you'd get to if we were at the Masonic Lodge, then hang around. I'm happy to be here. So
Yeah,
thank you,
like
everyone.
there's cookies at the Masonic Lodge, and,
Right.
yeah.
Well, we hope in January, we're going to give it a try.
Oh, good for you. Okay, that sounds good. Yeah.
And thank you, Eric. It was great to see you.
Yeah, good to see everybody. Thanks for having me. I'm going to leave my shed and go inside and
Okay. Good night.
eat the rest of my
I'm
dinner.
also going to leave campus finally, I think.
Yeah, I saw you in the lab there.
I don't trust my internet anymore.
How far up Neelan do you live?
I'm at the top. I'm on Berry Ridge, so past the school. It's 45 minutes from Arcata, so it's not too bad. It's pretty up
Okay.
there.
Well, drive safe.
Thank you. I will. Thank you
Okay,
all so much.
Thank you, Sarah. Good night.
Have a good night.
Okay.
Yes, thanks for
Okay.
having us. Good night.
Thank you, Sophia. Very good job.
Yeah, it was nice seeing everyone's faces, putting faces to the names.
Yeah. Yeah, maybe if I see you on the street, I might recognize you.
It's hard with the Zoom still. All
Yeah,
right. Have a good evening.
good night. So Donna, we got one name for the potential distant future speaker.
Eric?
Yeah,
Okay.
yeah. He's a good one to check back in with now and then.
You know, if it's just you and me here, I mean, I don't know if Gary and the other person here are.
Yeah, Jane and I are here.
Hi, Gary.
Hi, Carol.
Hi, Carol.
And hi, Jane.
Is that really Serena McNeil? Oh, she just went. Oh, she's a really good friend of mine since the 70s. My goodness. She just
Oh.
was a teacher.
Oh,
Yeah.
there.
That's great. I didn't know she's interested in plants. Wow.
Oh,
Did you know that about Serena?
yeah. I've been thinking about asking. I didn't really have a question, but I'm sorry that Megan left. I was starting to speculate about the little tongue-like structure on the leaf. Maybe it shades the opening to make it less obvious to the insects inside.
For Darlingtonia.
Yeah, for the Darlingtonia.
Well, do you know John Roth, who does a lot of interpretation up at Oregon Caves and around there and with the Siskiyou Field Institute?
I'm familiar with the name.
Okay, well, I went on one walk with him, and that when he looked at the Darlingtonia, he said that that the plant secretes some kind of insect attracting thing on the mustache and along the flange that goes up the back of the leaf and around, and that attracts the insects, and then the insects will follow it up, and following it up the mustache ends up in the mouth.
Oh,
Now,
okay.
maybe that was all just hypothesis, because
I've
ahead,
read in one book that it's called the fishtail nectary, which would validate what you just said, Carol.
yeah,
Yeah. Wow.
if it's a nectary, that's a pretty clear function.
Yeah,
Yeah, Jane has a really sensitive sense of smell,
I
so
do.
we'll have to see if it can be noticeable.
We will test that tomorrow.
Will you be seeing Darlingtonia tomorrow?
Our friend Catherine gave
Yeah,
Gary
we were given one.
from
I'm
the
gonna try
native
to.
plant
Oh,
sale.
okay.
Gary, a rather sizable Darlingtonia.
Well,
I mean, it's a good start.
yeah, it's a good. Yeah, and I'm trying to figure out just the perfect way to, to find a microhabitat for it.
We should go by our creek.
Well, I was thinking maybe underneath. Well, I don't know if it would get enough sun, but I was thinking that we have a perennial drip near the bird bath, and that has some potential to maybe.
Yeah.
But
Might
Yeah.
not get enough sun.
yeah,
Might not get
yeah,
enough sun.
the
honey.
sun is lacking there.
Yeah, well, let's see. Karenisa has Darlingtonia in pots at her house, and she definitely has limited sunshine. So it might not bloom, or it might not grow as fast, but I think it's got pretty broad tolerances.
Yeah,
Well-
I'd like to find there's just the perfect spot for it where it'd be really happy and do stuff.
Have you, Carol, personally been to the sites? I know one of them you've been to for sure because we've been there with you, but have you been to the other two sites she mentioned? In Gasky and
Well, Gasky for sure we've been to. I have not been to the place by Quincy.
Mhm.
And I'm not sure that the place she called the Mount Shasta place.
Yeah, that was intriguing.
Somebody asked, was that at Deadfall? And that to me means Deadfall Lakes. And I've been to Deadfall Lakes, but the dot on her map was not where I think Deadfall Lakes is.
right.
Yeah.
So I
Do you
don't
know of any locations of it within Humboldt County?
know.
That would be interesting to discover something like that.
Wouldn't
Yeah.
it? Yeah, no, why not at Horse Mountain? I mean,
I
there's
know,
water.
I know.
and-
It seems like a horse
Yeah,
mountain would be
around
perfect.
that north side there. It looks like it would be really good.
Yeah. Maybe
Well.
we should put some there.
I was just saying that, Carol. I was going to say that. And I go, oh my Jane. I was thinking we should sneak some up there and just start a patch. But yeah, the botanical area is really nice at Horse Mountain.
It is. It's
I
got
really
some
like that they
cool
close
stuff.
the gate and they don't, well, I can't say for sure it's closed right this minute, but it's usually
Yeah, at
closed.
least usually during wet weather.
Yeah.
Yeah, they close it in like, when the first rains come in the fall, they close the
Okay.
Port Orford Cedar gates.
Mm. So it's
So
to
Donna, what
protect-
was on your mind?
Well, this is just garden club stuff, so I don't know if Gary and Jane are
Oh,
interested.
that's fine. Yeah.
Well, if they want to
Fine.
leave, they can leave. Or
That's right.
Well, did you know that Mary Lou Goodwin passed?
No.
Yeah,
Oh.
she died
Oh,
3 or 4 weeks ago.
well,
Mhm.
she has a big legacy.
Yeah, she does.
But thank you for telling me.
And I'm sure you heard about Rick Tolley.
I did. I read
Yeah.
that on my North Coast Regional Land Tract.
Ollie.
Totally.
Yeah,
Yeah.
the artist.
The other thing that's on my mind, so my talk to the garden club in Fortuna was rescheduled from this last January till next January, which is now next month.
Right.
And I've just been thinking that I'm going to cancel it because they are meeting in person. They have been meeting in person, according to my contact there, for 3 months. And this is the same time that our county health director, Dr. Hoffman, has noted an especial surge in Fortuna and surrounding areas.
Mhm.
And I'm very concerned that with the holidays, Thanksgiving and Christmas coming up, there's going to be another surge. Our COVID rate today in the county was 35.
Speaker.
Hasn't been below 20 per day for months, as far as I can remember. And I'm just not at this point comfortable with the idea of walking into that room with people that are unmasked. And
Oh,
I had
goodness.
asked, I had asked Leanne, I said, so do people wear masks in the programs? I mean, I was astounded to hear they were meeting in person for 3 months already. And she said, well, most of them come into the building with masks and then eventually they take them off. And I feel really bad about canceling. It's only almost hardly a month away. And I know that this is a talk that people need to hear, but I am just more and more feeling like I don't want to be there. So
Yeah, that
But
sounds like
It
the
sounds like a
wise.
reasonable on you're the reasonable one.
Yeah, I think we all are, we're inclined to side with you on that.
Yeah, yeah.
Yes.
I mean,
But
we've
I'm
gotten this far.
wondering-
It'd be really
Wondering,
a shame
could
to
you-
get careless at this point.
I'm wondering if you could record your talk and give it to them and they could project it in some way. Do they have somebody who could do that?
I think they probably would. My thought was, I had another thought that I could do a Zoom presentation, but then I remembered that she said they don't do Zoom because they have a lot of older members that don't even have computers, much less are Zoom capable. So, but recording it, that's a possibility.
Yeah, I mean, bringing older members together in person in a building is probably not the best idea, especially.
Jeez.
Well, and
Yeah.
probably you've seen in the last week, there's been analysis across the country of counties that voted heavily for Trump, and they have the highest COVID rates and the highest COVID death rates. And in Humboldt County, I know Trump is a big thing in Fortuna. And and I just before the program tonight, I looked on the Eureka Sequoia Garden Club, and they're still meeting on Zoom. So Fortuna meeting in person is just suddenly becoming really scary to me.
Yeah.
You have a right to protect yourself, and it's your life. This isn't like a casual thing. So, you know, pre-recording yourself would be a really good idea, I think.
And how would I go about doing that? I am not really tech savvy.
Well, I can tell you what CJ did to present a talk at the meeting of the New Zealand Ecological Society 4 days after Thanksgiving. He was required to submit a recorded Zoom presentation beforehand, so that if it came to the actual time of the presentation that the internet connection was unstable and everything, they could still project what you had sent them.
Right.
So he figured out how to record the Zoom presentation as an MP4 or something.
Yeah. Yeah.
And then send the MP4 to their organizer.
Wow. Okay.
So the Fortuna Club would need somebody who knew what to do with these things.
Yeah.
Yeah, he basically did a PowerPoint and recorded it and turned it into a movie. And so, yeah, and there was a specific app that helped with that, right?
So
yeah, I mean, he mentioned it to me when it.
Here's CJ.
Okay, well, you just do your.
Oh, there you are.
There's hi, CJ.
Hi. You do your PowerPoint presentation. Okay, you have that ready.
This is a box of paper.
You do it on Zoom?
And then you do it on Zoom just yourself. Anybody else?
Can't hear you.
You do your PowerPoint presentation, and then you get on Zoom and share your screen on Zoom, just with yourself on Zoom. And I can send you the instructions they gave. It is a little challenging, but it's quite possible.
Okay, well, thank you. I will try to get in touch with my contact at the garden club tomorrow and ask her if she thinks that's a possibility. I have a feeling she'll say no, but it's worth a try.
Well, you've had this talk ready to give for so long, and it's such a good talk.
Well,
I love the topic.
Yeah, I do too. But I've had more trouble getting pictures from Pete.
Oh, yeah?
Yeah, big trouble. And he sends them. I said, just send your pictures as an attachment to the email, but he sends them in the body of the email, and I cannot extract them from there and get them into my picture file. And so I told him that wasn't going to work. So then just yesterday, he sent me all these pictures again, but they're all in zip format and I cannot unzip them. So I told him I couldn't do that. And so he said he was going to rest his brain for a week and get back to me later. So not making a lot of headway there. I mean, he has great pictures, but
Well, I'm remembering the talk on Forest Highway 1. Pete contributed to that.
right.
I guess I'd have to, I don't remember how he's provided the photos or how we got them.
Well, anyway, that's my current frustration.
So did we decide that you could give that talk to CMPS?
No, we didn't talk about that, but I'm going to give it at the Botanical Garden. You know, a version of it, probably similar, very similar. And I'm going to give that on March 26th.
Well, would you want to be in consideration for our May evening program?
You mean if you can't get Dr. Smith?
Yeah, I can't. Well, if we had you as a good backup, we could wait till later. Yeah, and ask him again.
I just don't feel like this is really the way I've structured it. It's really aimed at birders or gardeners, and it's nothing that people that are familiar with Talamy and the Talamy program we did and the Talamy books and the book reviews I've written, it's nothing new from that. So I don't know. I don't think it would be the best program.
Okay.
But anyway, I'm glad to have an opportunity because I've just suddenly just within the last few days become really nervous about the Fortuna situation. And I hate to back out on them, but I don't think I would feel comfortable. It's going to be indoors. It's going to be cold. They're not going to want to have a lot of ventilation. And if I'm surrounded by people inside that are not wearing masks, I'm going to be uncomfortable.
If they promised to wear masks, would it change the situation?
That's true. They're asking to wear masks.
I don't know. How would you feel?
And
I know I actually canceled. Let's see, am I muted? No. I actually canceled because I had offered to lead an Audubon walk. It was going to be this last week, and at some point I realized that the protocols had changed and it wasn't requiring either vaccination or masks. And so, you know, I had to have someone else take it over. And even, you know, because even outdoors, I think for, you know, you're going to be gathering close to people, you know, for
right.
And I think that certainly requiring vaccination is a reasonable thing to do. And
we do folk dancing with this group of about 30 people, and we require that everybody has proof of current vaccination, including booster status, you know, so they're on that path.
Right.
And then we have rules about no food and how far, like not holding hands. And so, you know, and everybody has to wear masks, from no ifs, ands, or buts about it. But showing that proof, would you be comfortable in that kind of situation? Maybe you can ask for that.
You know, Fortuna, this is a guess, but I would guess that they're not going to want to look for vaccination status. I mean, our county supervisor for Southern Humboldt has refused to tell if she's vaccinated or not.
Yeah.
So, yeah. So that's our county supervisor.
You have to follow your own instincts in these situations. And I think that if you're feeling this strong right now, then going at all, I think they need to hear that too from you.
Yeah.
It's kind of an education thing. Like, perhaps a different opinion.
Yeah. Yeah. Thank you.
It's a hard decision for you. I can see that. I can hear that in your voice.
But I certainly think offering to do it as a Zoom with, you know, with live question and answer is a very viable option, in these times.
Well, and maybe by now they figured out how to do Zoom. I don't know. But anyway, I really appreciate all your folks' responses. That's helping to clarify my thinking. Thank you.
Okay,
You're welcome.
yeah, too bad, Donna.
Good seeing you all.
Yeah.
All right,
Yeah.
I'm gonna say
Are
goodnight.
we the
so
only
thank
ones
you.
here? Because CJ and Carol, are we the only ones left? Yeah?
Yeah,
Sure. I don't want to have fun. We're wondering about the Christmas bird count on January 2nd.
CJ's in charge of that.
No.
Okay.
Doing it with you guys again.
Which count is that?
You asked the question?
Yeah. Well, we need to try to coordinate getting together and anyway. And we can go over all the Christmas bird count options. Again, might as well make the most about being, stuck here in the winter, right?
Yeah, what one is on January 2nd?
That's the Centerville.
No. Is it? Okay.
It should be.
I don't know.
That's it's usually the last Sunday of the period, and that's about the traditional time.
I want to make sure that.
Is CJ checking the
Yeah.
Audubon site?
No, I was checking. Well, I didn't write it on my calendar, so.
We just did today, CJ.
Okay, and it's the second, huh?
Yeah. We have to double check. The
Sunday.
Audubon site. Yeah.
Well, great.
It'll be fun to join you for that.
Yeah, I'm not
Yeah.
sure. Do you know who's organizing it? Or compiling it?
John McAllister.
Okay.
Well, I've written it down in pencil.
Sounds good.
Yeah, I tend to put things down, put things in my phone in electrons, which are just as erasable.
And they also evaporate, in my experience.
Yes. TJ, how'd your talk go? The one that you were talking about when we were there.
Well, I don't know. I haven't listened to it. Carol, how did it go?
It went fine. It was good. There were good.
You can't hear it.
There were good comments.
Wait, I think you're moved away from your speaker.
Is this better?
Yes.
Yes.
Okay. It went really well. There were good.
Great.
There were good questions, and in the chat there were lots of good comments. Some from important people.
I thought I did very good. Not world class, but very good.
All right.
Thanks to Carol, Christina, Duncan,
Yeah.
everybody here for Thanksgiving pulled together to, well, getting it recorded was a real problem. I didn't tell them. But now I know how to do it. Like somebody wants to do it, I can help.
Now you'll teach everybody else.
Yeah,
Huh?
that's good. You probably have some good advice to offer for some of the Godwit Days ones. 'Cause I know we learned a lot last year, but one of the things we learned was there's a lot to learn.
Yeah. Well, I've got all the t-shirts laid out here. I'm working on putting them together.
Oh.
Can you see a picture of it?
Yeah, just moving the camera.
Oh my gosh.
Wow.
Oh, that's so beautiful. Oh, wow. How'd you make a decision about which one goes where?
Well,
Oh, that was easy.
Maybe that was easy.
Did chronologically.
Oh, chronologically. I was gonna say by color, but duh. So it starts up at the top and goes that way, right? I'm thinking of.
Yeah.
Yeah. It starts at the top. So how many years is that, CJ? Gary?
It's
You could count them, I guess. Wow.
yeah. It was,
My goodness. So who's this quilt going to, CJ? Gary?
But have more than one color t-shirt.
wait, I'm not here. No, we're not hearing you. Yeah, I think your mic is directional.
Yeah, it must be. That looks-
If you look at, there's some shirts that have two colors on them, like the rail.
Uh-huh. Oh, right. So you were just deciding which one to put in?
Well, I started to, but I need somebody that has a good color sense. They're like, so we'll see.
Yeah, well, I'm always happy to throw my 2 cents in, but
What are you doing tomorrow morning?
oh,
We don't have to go
Tomorrow
into
morning.
Rico.
yeah,
We're gonna do my glasses, but I can call about it tomorrow. Go
yeah,
over- Let's do it tomorrow
Yeah.
morning. Go tomorrow morning.
okay, yeah, I mean we can work that out.
Yeah,
Yeah,
my glasses can wait. This is a project that, so who's gonna be the who gets to benefit from this quilt?
Me.
Wow, it's about time. Help your, that'll be the most amazing quilt. You guys have to get pictures.
Oh, Gary, would there be some time tomorrow you could come over?
Yeah.
Carol claims it.
yeah.
I'm gonna